So you have my screen now?
Yeah, we can see it.
All right. So today I'm planning to talk about stereochemistry of metabolites and what can be called chiral metabolomics. Uh, chirality is an old topic, maybe as old as the new concept of chemistry, starting in 1812 by Jean-Baptiste in France, who discovered the rotation of sugars when they are placed in a polarized light environment. Later in 1848, Louis Pasteur discovered the same thing with tartaric acid, and he understood that tartaric acid enantiomers have different crystalline structures and can rotate polarized light in different ways. So, and he understood that this is because of the asymmetry of its molecules. So as you all know, chirality is a concept of having four different functional groups on one carbon structure. And we will see how and why this is important to us in metabolomics. So one of the oldest concepts of metabolomics has been known for plant essential oils. So we all know that different plant essential oils like menthol, isomenthol, neomenthol, or neoisomenthol have different kind of enantiomers. They might be positive enantiomers or negative enantiomers, or R enantiomers, S enantiomers, just different kind of nomenclature. And also the other thing which might be interesting to note is in most of the cases, different different physical properties, actually different biological properties like taste or odor are different. So asparagine has a bitter taste on its S enantiomers, while its R enantiomer has sweet taste. And limonene has citron taste on its L enantiomer, while D enantiomer has orange taste. So this means that the enantiomeric composition have some biological effects. I'm just trying to show some more examples. So let's have some more important example like THC derivative of cannabis. So tetrahydrocannabinols are actually four enantiomers, and these enantiomers have important properties based on their activities, and this is actually why some of these cannabis species are more expensive. Some of them are not, because actually the enantiomer composition of these important derivatives are changing. So historically, there was a misunderstanding of the chemical purity of natural products, and that was natural products are always biosynthesized enantiomer specifically. This is actually false, and especially in recent years, I would say recent 15 or 10 years, this has been greatly challenged by many researchers, and now we can understand why this is happening. So there are some reasons described why this is happening, and we know that like enantiomers of metabolite are racemized due to storage in different cell compartments, especially the pH of that compartment. Enantiomers of metabolites are differently biosynthesized in different species of the same plant due to isoenzymes potentially. We have some enzymes which are not enantiomer specific as much as they should be. And the other thing is a huge number of natural products are being racemized by the action of enzymes like racemases or while trafficking in the cells. All these phenomena make a biological system and a metabolic system not to be enantiomerically pure. So let's have some examples on this. In the biosynthesis of terpenes, especially monoterpenes, we know that everything starts from geranyl pyrophosphate. On the top, you see GPP. So it has a pyrophosphate moiety, which can actually just move around. It can fold just simply on the right side or the left side without any interaction with any enzyme. So what happens if it folds on the right side? We will have positive alpha-pinene or positive bornyl pyrophosphate. If it folds on the right side, we will have negative pinene metabolite. And also we might have positive limonene R enantiomers or S limonene. So this shows even sometimes we have some unbiological phenomena to make different enantiomers. Another interesting example can be carbone. We know that carbone is mainly found in peppermint. So the R enantiomer carbone is responsible for the taste of mint actually, but the S enantiomer is the responsible for the taste of caraway. So it's actually just found in caraway seeds. But what might be more important to us is their physiological activity. As you see on the image below this slide, the S enantiomer carbone has hypertension effect, has many effects on brain activities and on hepatic metabolism, while the R enantiomer has effects on microorganisms, muscular contraction, and tumors. Both together have similar effects on reactive oxygen species and inflammations. So we can have some more examples on drugs which are much more important economically. And here I have a list of couple drugs from thousands of drugs whose R and S enantiomers are pharmacologically different. You can see thalidomide, ibuprofen, naproxen, aflatoxin, L-DOPA, citalopram, many of the drugs we might continually sometimes use. So most of them have different enantiomers with different activities. Let's have some other examples, especially in plants. So here we have gossypol terpenoid in cotton seeds. So gossypol actually has no central carbon, asymmetric carbon, but as you see, it has two big halves. And the problem is these big parts make some rotation restrictions over the carbon bond. So different enantiomers are being made by this action in cotton seeds. So positive gossypol is a toxic enantiomer and is being used in anti-tumor, antiviral activities, while the negative isomer is being very harmful for livestock because it has a spermatozoid motility and activation of sperm enzymes. And so this is actually why it is being discovered. Some other interesting examples can be quinine and quinidine, which are produced from American cinchona tree. On the left hand, you see the molecule quinine, which is known for its anti-malaria activities, while on the right hand, you see enantiomer quinidine, which is used as an anti-arrhythmic drug. So you see two different enantiomers have totally different activities. And let's have a look at Planck signaling systems. As you know, acetic acid, jasminic acids, and many of these signaling molecules have enantiomers. As you see here, they have huge effects on gene expression and phenotypes. And in this paper, you can see R acetic acid or S acetic acid have totally different effects on the gene expressions and even on the growth and development and phenotype of the plant. Here's just a beautiful list of the metabolites, just to see that on the top we have versicolumide B as an alkali produced by aspergillus families. So you see different aspergillus families produce different enantiomers. So this is pretty interesting that the same organism but different species makes different enantiomers. And you see R-limonene is being made by citrus plant, while S-limonene is being made by Mentha. So on the below side, on the bottom, you can see reticulin, which is a narcotic alkaloid. So you can see that papaver somniferum produces positive reticulin enantiomer, while the Rhamnia culture produces the minus enantiomer. So let's have a look at the amino acids, and we will see why this is very important for us, especially for biomarker studying cancer. So in bacterial communities, the amino acids are being used, especially in dearginine and D-lysine ones, as a signaling repellency molecule. So whenever there is an unfavorable place like heat or salinity in water, lots of dearginine or D-lysine would be metabolized to signal the community to move, to run away. And in cancer, you can see that in cancer like gastric cancer, even though in gastric juice or saliva, different amino acids like D-alanine, D-proline, D-serine are being highly upregulated compared to control ones, and also in hepatocellular carcinoma, the same thing can be seen in the serum levels of the amino acids. So the amino acids and cancer, actually, if you search about this topic, you can see many papers, especially in very high-ranked journals. At least I have a couple papers in Nature, and you see huge interest has been paid to this issue in recent years. So the amino acids are being important biomarkers for cancer, especially for modulation of cancer proliferations, or even as a biomarker and their accumulation in breast cancer and in some other ways. So I just brought one example in here, and you can see 2-hydroxyglutaric acid, which has been shown in many different types of cancer. So here you can see on the heat map that D or L enantiomer of hydroxyglutaric acid has totally different effect on reprogramming of amino acids in the plant, in the cancer cell, colorectal cancer cell. So this obviously shows that different cancers have different responses to enantiospecificity of their metabolite, especially at the metabolic level. So the story of enantiomers, the pharmaceutical industry started with a disaster actually. In 1962, thalidomide was being used as a sedative for pregnant women, but the problem was at that time there was no idea about the importance of an anti-purity of drugs in the pharmaceutical industry. So the problem was only R-thalidomide was the sedative drug, while the other S enantiomer was a teratogenic. So up to now, there are tens of thousands of people around the world who have malformed limbs. Yep. So why is chirality important in industry? So at this time, 56% of drugs are currently chiral products. And today, $70 billion is the world market of chemicals, and it's supposed to reach 160 million in the next 10 years. $160 billion actually by an annual growth rate of 10% per year. So having a look at the final formulation worldwide sale for enantiomer products, we will see that in 2005, a total of $225 billion of chiral drugs have been sold in pharmaceutical industry. So this obviously shows how big and how important this market is. So let's have some other things which might be more important to us in here. The global market of D amino acids is around $20 million at this time. And some other analytical things is the global market for chiral chromatography columns, which is 80 million just right now. So what is chiral chromatography? So to separate different enantiomers as they have totally similar physical or chemical properties, we need to immobilize a chiral selector on the surface of silica gel in the HPLC column. So this is done by a chemical reaction. And what happens afterward is when the racemic molecule is passing through the column, there will be different interactions between two enantiomers with the chiral column. So there will be different retention times and they will be separated. So one of the things I did, I would say 15 years ago or 16 years ago when I was a master's student was making vancomycin-based microcolumn for separation of drugs for some industrial projects. So on the right upper hand, you can see the reaction we made, so which was a special organic reaction to immobilize vancomycin through one of its functional groups to silica gel particles. So, and then it was packed into a column by a pneumatic pump at very huge pressure, like 40,000 psi. So on the bottom right side, you will see the column I made, which is actually a micro column. So this column was packed by that instrument and the pneumatic pump we had. And on the bottom left hand, you can see some of the results of the works we did, the separation of different enantiomers of natural products and drugs. The other things we continuously did was developing some tapered optical fiber-based biosensors for the same purpose. So in the yellow image in this slide, you can see the picture of a fiber optic, which is actually tapered. You see the narrow side of the fiber optic, which is produced by removing the upper layer of the fiber optic. So the narrow part will be our reaction medium, the sensor medium, which is placed into the upper side of the image. As you see the red cell, which is our fiber holder. So this sensor was placed into a laser system and could detect analysis at like nanomolar level, very sensitive ones. And we try to do the analysis, the interaction between different molecules, different enantiomers and sugars, or between some epimers, some DNA-based biosensors. And I just like to tell about a little bit more about the detection system of this laser-based sensor. So the detection system was actually a refractive index system, a very sensitive one. And based on the molecular interaction, there will be a chemical shift in the signal of the refractive index detector. And that was the signal which we recorded and analyzed the result of the study. Later on, I moved toward the study of metabolism of amino acids in the plant systems and biological pathways. So historically, it is believed that the amino acids kill plant, as you see in this picture on the upper beautiful plant. You can see that these plants are treated with D-serine and D-alanine, but the fact is they are actually mutant plant with the amino acid oxidase genes, so they can metabolize the amino acids and they can survive. And on the bottom side, you can see the wild type plant, which are actually dead because of the treatment of the amino acids. What I did was a study of ornithine metabolism in plants. So ornithine is a central amino acid which can pass to two competitive by pathways. On the left side, it can go to nicotine biosynthesis. On the right side, which is common with animal cell and cancerous cell, it produces polyamines like putrescine, spermidine, and spermine. So I use the plant cell culture as the platform to study metabolism. As you see in this picture, we have different L enantiomers of ornithine or D enantiomers and compared them with control sample. We use a bunch of metabolomic works. I guess I did this project in 2010 when I was a PhD student in Metabolomics Australia, and we had access to different kind of platforms like LCQ Top Metabolomics for untargeted analysis, GCMS-based metabolomics for sugar and hydroxy acids, LCMRM-based LC-triple quad actually for amine and amino acid analysis, and I also did some PCR work for gene expressions. So the first interesting phenotype we observed was the viability of the plant. So we actually observed something just opposite what we expected. So on the right hand, you see the L-ornithine treated plant cells, which are dead. As you see the blue color, meaning they are dead cells, but the D-ornithine treatment did not kill the cell. So we try to understand what's happening. So from the metabolomics works, I just telling you about the MRM LC-throughput quad work. So we developed an AQC-based derivatization of amines, which produce a specific derivative of amines, and it was dissociated in the mass spectrometer. So later on, it was passed through the second mass spectrometer, producing an AMQ metabolite at the molecular weight of 171, which could be monitored as the mass loss in the mass spectrometer. So an MRM analysis was performed, and we were successful to do the metabolic changes of all the metabolites. And I want to talk a little bit more about this thing. So the interesting thing we found was only arginine was hugely accumulated in the plant cell. So as you see, arginine is accumulated up to 80 times compared to control content of arginine. The fact is arginine is a $500 million amino acid in the market. So it was interesting that by this simple treatment of D-ornithine to the plant cells, we had 80 times, 80 times increase in the upregulation of arginine in the plant cell. So we try to see if this is L-arginine or D-arginine. So here are the spectrum of the standards injected to mass spectrometer. When we analyze our molecules, our sample, we are understood that 100% of what produced in the plant cell was only L-arginine. So we concluded that there is an unknown metabolic pathway. We never heard about this before. In that D-ornithine treatment to plant cells can produce L-arginine. And as you see, I did this with different, very old version of Metabolomics, I guess, at the time. And as you see, totally different separation between R and between different enantiomers of ornithine can be seen in my work. So at this time in TMEEC, I am doing some more analysis on a similar project to analyze to upregulate the level of scopolamine and hyoscyamine or atropine, which they are important drugs as well. So what I'm doing here by changing the regulation of those two amino acids up on the top, arginine and ornithine, and using their different enantiomers, I'm trying to see what's happening in the biological pathway of the plant, which is atropobelladonna plant, and on the final its precious metabolite, which are hyoscyamine and scopolamine. So let's go to chiral metabolomics and talk a little bit more about what it can be. So for chiral metabolomics, we can do two different kind of things. The first thing is to use chiral selectors for NMR and LCMS to make diastromers in the media in the matrix. The second thing is using 2D GC or 2D LCs, in which the first column is a chiral column and the second one is a chiral column. So here you can see a chiral GC work of an analysis of amino acid. As you see, this is an amino acid sample, maybe from serum or something else. And a very nice GC experiment has been done, a chiral GC, and all the amino acids are very nicely separated in less than like 35 minutes. The same thing can be done by LC. So by LC, probably it's better to use LCs, where the first column can pick up the metabolite we want to analyze for the enantiopurity and send it to the second chiral column. So by this process, we can have continuously switching between different columns, the first column and second column, and send the selected metabolite to the second column to study its enantiopurity. So here is the NMR works done. I have to be very careful with my words here, I know, because I'm not an NMR specialist, but I would like to show these beautiful papers. As you see in these papers, the NMR discrimination of an amino acid matrix with L and D enantiomers is done by parahydrogen-induced hyperpolarizations. So as you see, an iridium chiral selector was added to the solution, and by making different diastromers and a parahydrogen-induced hyperpolarization in the instruments, different metabolites can be very nicely analyzed in this in 2D NMR. And also I would like to talk about benchtop NMR, which I think would be very beneficial both in chiral analysis or in metabolomics. We all know that at this time, many clients are trying to do real-time monitoring. They like to have simple instrument to do some metabolomic analysis, and there are many projects demanding this concept. We never have a portable mass spectrometer, a portable GC or HPLC, but recent advancement in benchtop NMR has some promising news about this. So I collected a list of 6 producers of benchtop NMRs in the market. As you see, Bruker, Magritech, Nanalysis, Oxford, Thermo Fisher. We are producing different kind of benchtop NMRs from 84 megahertz to 125 megahertz. Interestingly, some of them are very lightweight ones, up to 55 kilogram, which is very nice for having portable NMRs. And definitely, I know that the line-based sensitivity and resolution of these instruments should be considered. But here we have some authors who claim that they can do some considerable works by these very top NMRs compared to very expensive ones. So in this paper, on the top, you can see the result of a 700 megahertz Bruker instruments, results of tuberculosis patients, and on the blue one, you can see result of an 80 megahertz magnetic NMR, and the result is actually there might be some modification, some result as a metabolic profiling of the problem. And in this paper, you can see analysis of type 2 diabetics. So on the top red, you can see the profile of a diabetic sample, and on the bottom blue one, you can see the result of a healthy control ones. It's done on a 60 megahertz NMR spectrometer. So you can see that around 27 metabolites are being analyzed by this. I think this is pretty interesting that we have at least 27 metabolites identified, and here authors claim that actually in metabolomics we just want to see what's happening in the final part. So in the final part, we see the results are actually the same. So on the left side of this loading plot and score plot, you see a very nice separation between different groups, which belongs to a 400 megahertz NMR. And on the right hand, the score plot and loading plots of 60 megahertz. So you see actually the results are the same. So for some special cases, this might be interesting idea to explore, especially for biomedical applications or for some real-time application, especially when automation processes are developed to cover the low resolution, low sensitivity of these instruments. I think this would be a pretty interesting idea to explore for commercial uses of NMR, and this is definitely a field that GC and LC or mass-based work can never compete anymore. Thank you very much for your time, and I would be happy to have your comments.